Good morning. My name is Marina Good. I am from Fitchburg, Massachusetts, and I am actually a graduate of Montachusett. My experiment flew with Mission 7, and my research question was, how is the growth of the bacteria Rhizobium radiobacter affected by microgravity? Our team members, I was the principal investigator, my collaborator was Jezreel Bruno, and Paula DiDiego from Montachusett was my teacher facilitator. So our proposal, it was focused on how the growth of the bacterium R radiobacter and by extension the production of endotoxins could be affected by microgravity as opposed to its observed growth and endotoxin production on Earth. We predicted that the freeze-dried bacteria could be successfully activated and grown in microgravity, and that more bacterial growth and higher endotoxin production will occur as a result of microgravity. So this is our packaging and interactions. We had a type 3 FME mini labs. That means we had 3 different volumes. Our first was the freeze-dried bacteria, and then we had a growth media. We used luria broth, and we had a growth inhibitor called acetosyringin. So 5 days before undocking, that was May 7th, we activated our experiment and mixed the bacteria and growth media. And then 2 days before undocking, we opened the second clamp, and we deactivated the experiment using the growth inhibitor. So we performed an optical density test using the SuperNate. We did a microscopy analysis. We did a mass pellet test, and we also did an endotoxin limulus amoebocyte lysate biological assay. So for our endotoxin biological assay, we used endotoxins extracted from the cell membrane as well as those found in the SuperNate. So these are our results for the optical density. And just so you guys know, an optical density test is basically shining light through something, some kind of a substance, and you're measuring the amount of light blocked by substances also in the liquid usually. So we ended up having an average optical density for our ground truth of 0.386 and an average optical density for our flown sample of 0.39. So not very statistically significant, but definitely a difference. So we were also worried about the luria broth that we grew our bacteria in. It did slightly discolor the samples, so we subtracted the background absorbance, so whatever discoloration, we subtracted that from the original optical densities. So this means that based on these observations, there were virtually the same amount of cells and growth present in both samples, although the flown sample had a slightly higher average optical density. And then for our microscopy, we found that in the ground truth, there were more compact groupings of bacterial colonies, while those of the flown sample were slightly more spread out. And the cells of both samples were rod-shaped, as we used a Bacillus bacteria, but the cells of the flown sample were slightly more rounded, and then the cells of the ground truth were more distinctly rod-shaped. And then the results for our mass pellet test. So a mass pellet is where you take a sample and you centrifuge it. So you end up having a separation. You have a liquid with typically a solid that precipitates out, and you take the mass of that pellet after drawing off the liquid. So we found that there was a significant difference between the mass of the ground truth versus the flown, as you can see. We had 0.01726 grams as an average mass for our ground truth, and 0.01244 grams for our flown samples. This difference was unexpected, as we thought that the ground truth would be lower instead of higher. This could be due to human error, though, as we could have left some SuperNate left in the tube as we were drawing off. For our biological endotoxin assay, basically this is just a detection used to look at a chemical that is produced by a bacteria. So endotoxins can either be secreted into the SuperNate or into the surrounding environment, or held in the cell membrane, and you have to lyse the cells open and extract it. So we did both. We looked at both. We did confirm the presence of endotoxins, as there was a color change in our protocol, and we used a high detection test because our sample, when it came back, was very, very cloudy. So we thought that there would be a really high amount of growth and therefore higher amounts of endotoxins. This is a picture of our samples incubating in a hot water bath as per kit protocol. This limulus amoebocyte lysate is actually extracted from horseshoe crab blood, and it has the ability to bind to this endotoxin, and it turns it pink. So I thought that was pretty cool. And then this is, we also ran an optical density test with this assay. It's part of the procedure. We use standards just to generate a standard curve. Those are on the left, and on the right is our table and results. So the average optical density from our flown supernatant, and keep in mind it's diluted because the supernate dilutes the endotoxins. So we had a 1.153 repeating optical density, and then for the ground truth it was 1.12267. And then for the extracted endotoxins from the flown sample, we had 1.099. And then for the extracted endotoxins from the ground truth, we had 1.056. And these are in endotoxin units per milliliter. And these are our more results. You can see the standard curve on the right. And then on the left, these were the actual endotoxin units per milliliter that we tested. And this was the most significant results that we had. For the endotoxin concentration of the ground extract, we had 0.275 endotoxin units per milliliter. And then for the flown extract, we had 0.287 endotoxin units per milliliter, so definitely higher. And then for our dilute, we had, from the ground, we had 0.293. And then for the flown dilute, we had 0.302. So the knowledge gained. The optical density of the flown supernate was slightly higher than that of the ground truth, but not remarkably so. The bacterial colonies of the flown sample were not as compactly grouped as those of the ground truth. The bacteria of the flown sample appeared slightly rounded in shape, while the bacteria of the ground truth were more distinctly rod-shaped. The mass of the flown sample pellet was slightly lower than that of the ground truth pellet, but not remarkably so. And the endotoxin concentration of the flown sample, both the diluted supernate and the endotoxins extracted from the membrane, was higher than that of the ground truth in both the undiluted sample and the extracted sample. So I came up with a couple questions. I'm not going to read them all. I know I'm running out of time. Most of them had to do with observed changes and differences in both the ground and the flown truth. These are our mentors. Thank you to Dr. Kristen DeAngelis from the University of Massachusetts Amherst in the microbiology lab. And thank you so much to the doctoral candidate Gina Shaput, also from the University of Massachusetts Amherst in the microbiology lab. Both were very helpful. These are our sponsors. And any questions?
Do we have any questions?
Tim Livengood, NCSE. So how did you choose this particular organism?
So originally, when I was working with Jezreel, I was a sophomore when we first started working on this experiment, and we were both very interested in cancer. We've both been affected by it in our families, as I'm sure everybody really has. So we were very interested in that, but you can't send anything human in origin up to the International Space Station for safety reasons. So we did some research on like a plant cancer, because that's not human, so you could send it up. So we ended up coming up with a bacteria. It was originally called Agrobacterium tumefaciens. That's what we studied actually in my biotechnology class my senior year. And it's a known vector for genetic modification. So I've always been very interested in genetics and genomics and modifications and things like that. So I thought that was pretty cool. And so the fact that it was a bacteria and we could fit it in a tube was just a plus. That's just awesome. So we ended up picking it basically because it's a plant cancer and it causes the uncontrolled cell growth, the development of a tumor, very much like a human cancer would.
Kirsten Larson. I'm from Seattle. What was the most surprising result to you from your experiment?
What I found most surprising, because in our hypothesis, we had predicted that the bacteria would grow exponentially in microgravity, as has been observed in other bacterial experiments. Ours did not appear to do that, but what Gina, our doctoral candidate that helped us a lot, was saying is that bacteria does grow exponentially. So it curves up, and then it tends to plateau, so it stabilizes, and then it starts to die. So what she was saying is that our bacteria very well could have grown exponentially in microgravity, but by the time we'd introduced the growth inhibitor after 3 days of activation, the bacteria had already started its decline. So when we cut it off, it wasn't all that significant as opposed to our ground truth. So we're not exactly sure. There's no way really to track it in this experiment, at least. But I think for me that was most surprising. I was hoping for there's really crazy numbers that were really high and just very, very exponential growth observed. So that was probably the most surprising for me.
Thank you.
You're welcome.
Thank you so much.
Thank you.